because we were asked to test the wheat side, and a whole lot of green beans and corn and pieces of crud dumped out of it. It was awful. So it's like a casserole, so it's a whale casserole. And this one here, I'll get you one. Anyone that'll add it? It's 2. Sits like that in the drawer. So just that bit showing.
Hippo.
So that's a lower incisor for a hippo. That's the reason they do so much damage. I think they kill more people in Africa than anywhere else. So hippo bottles. Well done. Okay. Enough of the good stuff. Okay. So just hang in with us for the next little bit, because this bit is a little bit tangential, but it'll actually mean something, I hope. What we've done is we've looked at what conservation genetics does, a bit about the different markers that are around, a bit about how we use them, etc. And what I want to do now is to concentrate a little bit on what actually changes the genetic diversity of a population. So if we can understand that we lose genetic variation when we don't have migration in a population, then when we're coming up with management plans or recovery plans, we can sit there and a part of that recovery plan might be that every 5 years we drop in another 10 individuals back into that population. In other words, artificially cause migration to basically increase the diversity again. In other words, if we understand what changes it, then we can perhaps understand how we can mitigate against them when we're bringing up management conservation. So I want to talk about these in terms of that, and then I want to talk a little bit about some examples about how we've used this stuff. So hopefully at the end it's a little bit more interesting, a little less dry. What I want to talk about first is migration. So migration is effectively a term that's to do with the movement of an individual either in or out of a population. And it essentially equals what they refer to as gene flow. And gene flow being the movement of genes from one population to another. It has 2 important effects. The 2 important effects is that it introduces and spreads unique alleles in new populations. So you imagine if you have migrants from a population and you have residents within that population, introducing migrants from a different population, because, and let's assume that originally they were all one population, similar to that Hardy-Weinberg Island sort of picture I had, is that you would have a genetically mixed population. A vicariant event comes through, something causes the population to become separate, and they'll go of different evolutionary trajectories, and at some point in time, the populations will be quite different from one another because things are driving them in different ways. So you can imagine you have this resident population, and you introduce migrants from a different population. What you will effectively be doing is introducing new and unique alleles back into that population, and the sum effect is that the population in the new one will actually have an increased amount of diversity, an increased amount of alleles as a result of that new material that you've introduced. And the second major effect is that it affects allele frequencies of the migrant and the recipient populations, particularly if they differ from one another. And it's effectively looking at the gene flow between the 2 populations. One of the things about migration is that change can happen very quickly as opposed to mutations. Another way of introducing new genetic material into a population is to wait for some mutations to arise in the population, and that will introduce new genetic variation that wasn't in there before. The reality is that if you look at the frequency of alleles and you look at the mutation, is that those changes take a very long time to actually occur. And when we do a lot of the modelling, we're talking about 40,000 generations for a population to increase or decrease, increase 90% of the diversity in the population because of mutation. In other words, we're going to be waiting a long time. The reality is, we know from conservation genetics or conservation in general biology, is that unfortunately populations are decreasing at a much rapid rate than they are actually increasing. So we're simply not going to have enough time to accumulate new genetic material as a result of mutation, because the declines or the rare occasions where populations are increasing occur basically too slowly for that to actually happen. Whereas if we were to look at changes in frequency as a result of migration, then it doesn't take very long, sort of 400 generations, to generate a bucket load of new genetic material into the population. I'm going to talk about this in a second. And what it does is it prevents allelic fixation. So if you've got populations that are very small, ultimately they will become genetically fixed because of genetic drift, and you'll only end up with one variant in the population. Introducing a new migrant introduces that new, it reinvigorates the population. So the gist of, I guess, what I'm trying to get to in this slide is that migration happens at a much faster rate than mutation in terms of introduction of new genetic material, if you've got it available to you. And this has a relationship, and I will talk a little bit about this, and we'll use this later today in these Northern Quals and in that assignment you're going to do. Is going to talk a little bit about what they refer to as FST values, or population differences. There's a relationship between FST and the migrants that occur in each generation. And the relationship being 1 on 4 times the number of migrants plus 1. And the relationship is this: if you look at FST, FST goes from 0 to 1, and it's a sliding measure of how different the population is, and it's a fixation index. And by that what I mean is if you had a single population and you had a genetically mixed population and they had exactly the same allele frequencies in those populations, you would have an FST of 0. In other words, there's no difference between this population and this one, because they're essentially exactly the same population. If you had 2 populations and in there you looked at the allele frequencies in population 1 and compared it to population 2, and all those alleles were completely different. In other words, there's no actual information that's identical or shared between the populations, then you have an FST value of 1. Make sense? Yeah. So an FST of 1 means they're very different. An FST of 0 means they're effectively a genetically panmictic population mixing. Lots of mixing happening. So let's assume we've got a population, 2 populations where they're effectively genetically completely unique. And then we look at the number of migrants per generation. As we increase the number of migrants in each generation, it doesn't take very long for that population to in fact become genetically very similar. In other words, 2 really unique populations, you introduce 1 migrant and all of a sudden those populations share 20% of their genetic material. And it doesn't take many more migrants after that till you're effectively down to this level where the 2 populations are effectively genetically the same. I'm starting to lose people with math formulas. The gist of it is, and it makes a lot of sense, you've got 2 genetically different populations, you start mixing them, they will start to resemble one another as those genes become accumulated in that new population. So it's a really useful and interesting statistic because it's been abused and used in a really constructive way in conservation biology. But FST is a statistic that tells us how different 2 populations are. And formally it tells that if there's a deficit of heterozygotes in the population. And as I said, ranges from 0 to 1. And Sewell Wright came up with these sort of metrics on the amount of FST versus how different a population is. So if you've got an FST of 1, it means the alternate populations have alleles that are fixed and completely different from one another. And a 0 means there's no genetic divergence, they're all genetically mixed up and panmictic. And these different levels are, little genetic differentiation, moderate genetic differentiation. This will be a bit more relevant when we start looking today at how different those populations are. It just gives you a metric on how different populations. There's some examples, and I'm not going to talk about them. I'm not going to get that in the exam, but some populations, for instance, humans have incredibly low FST values. What does that mean in the scheme of things? Very similar. So if we take, even though phenotypically we can get a very large amount of variation, we can have very different skin colours, we have very different tolerances, etc., etc. At the end of the day, we look at the underlying genetics underpinning human society, they have very, very low levels of differentiation, which means they're effectively mixing very large. What's really interesting in the last 100 years is that FST value has changed, and it's become even closer to 1 or 0. 0. We're actually becoming much more homogenous. So the ability to travel to different countries, the idea of intermarrying between people that we wouldn't normally marry with, for instance, etc., is changing. So the demographics of human society are changing their FST values. So humans, I guess the point of it is we have really, really very small levels of differentiation between populations or where we sample. And we can go up to some of these really weird stuff. So house mice, normal musculus, have 0.13. We go back to Sewell Wright's, it puts it in moderately genetically differentiated. So house mice have relatively large amounts of differentiation between them. And there's a whole range of different ones. Jumping rodents, so the Natomes mice, which are the little desert rat rodent things, have huge FSTs. So if we look at two different populations, one from Gibson Desert and the other one out in Strzelecki or whatever, they've got huge amounts of differences between them. In other words, there's not a lot of mixing between those mice populations. They're genetically being historically quite different from one another. Okay, and this is that graph, and it was used, as I said, in a very creative way in conservation biology. Because effectively, if we have an effective population size and a migration rate that's very small, then we would expect a very large FST. That makes sense? Small population, very little migration between populations, mean the FST will be closer to 1 than it would be to 0. If we have an effective population size and migration of less than 1, the FST is greater than 0.02. So that means if you know the FST and the effective population size, we can calculate an actual migration rate between populations. So we can use them as independent ways of working out how much migrations are happening between some of these fragmented populations. So if there is greater than one migrant per generation moving between populations, then Michael Sewell came up with this idea that by introducing a single individual into a population per generation will be sufficient to completely genetically mix a population back up. And it became sort of the gold standard in terms of translocation policy. So Parks and Wildlife, where they do translocations, use this one migrant per generation rule. It's been around for a while. It's been modified a few times, but it's basically hasn't changed a lot. Is that by introducing new migrants per generation will be sufficient to maintain diversity in the populations and won't lose them. Okay, so a little bit about some of the limitations of genetics and conservation biology. The actual area is relatively young. Conservation biology has only been around since the 1970s. Conservation genetics was sort of a spin-off of the theoretical side of genetics, sort of fly breeding and all that sort of stuff. And people realised they could actually use some of these concepts in applying them to endangered species work. And it's only sort of 30 years or so old. It's not particularly old science. Many of these techniques are, or were, relatively expensive. And so an honest project for me using microsatellites would be roughly costing about $10,000 to do that project. We can now sequence the entire genome of 100 individuals in the population for $5,000. And when you think about it, that's an enormous amount of data. So it's becoming a lot cheaper. So sequencing genomes is a very cheap thing to do now. You need to handle material properly, so it needs to be preserved properly. You need to be able to get it yourself. And at the end of the day, we wonder about whether genetics has any contribution. I have a good mate in DBCA called Andrew Burbidge, and Andrew argues with me all the time about the importance of genetics and conservation management. And the example he uses is this thing called the black-footed rock wallaby of Barrow Island. Barrow Island's a little island off the northwest of the Pilbara. It has a standing population of, I think, between 1 and 5,000 black-footed rock wallabies. These things here, they're an absolutely beautiful animal. They've been isolated between 5 and 10,000 years, so since the last glacial. So they've been out on that island marooned since time began. And at the end of the day, there's still bucket loads of them out there. A couple of thousand animals is not an insubstantial number of individuals. And Andrew is sort of arguing, well, why bother about genetics? Because there's bucket loads of them there, they've been there for 10,000 years and they seem fine. And he's got a good sort of point, because why haven't we, and this is the point I argue against him, is why haven't we found those rock wallabies on every other island of Western Australia? And I can tell you why. It's because there were rock wallabies on those islands at some stage when they were connected to the mainland. They were marooned, exactly the same Barrow Island was, but they've been through a bottleneck such that they've never recovered, so they've become extinct on particular islands. It just happens that Barrow Island's big enough to buffer them. And the reason he talks about the genetics is we looked at these populations of rock wallabies from Barrow Island, Exmouth, which is directly opposite on the mainland, and then a population down in the wheat belt. These are found in those wheat belt, isolated wheat belt colonies. And we looked again at microsatellite markers, and we looked at heterozygosity, in other words, one measure of genetic variation. And if we look at the wheat belt, they have about 60% genetic variation. X-mouth, they have about 60% genetic variation. And on Barrow Island, they have the lowest amount of genetic variation ever recorded in any mammal that's ever been measured. They have 5% genetic variation on Barrow Island. So even though there's lots of rock corbies out there, they're actually really, really low effective population size, and they have a very low genetic diversity, which is not uncommon on islands. We then started looking at a whole range of different things like hematocrit, which is looking at the amount of red pack cell, red volume, or red cells in their bodies. And the ones on Barrow Island had a very reduced amount of red cells. In other words, they were showing signs of anemia. They had very high parasitic tick loads in them. And if you look at the percent of females that are carrying pouch young, only about half of them on Barrow Island are carrying pouch young, whereas they're sort of like 80-90% on mainland sites. So they're also showing signs of inbreeding depression. In other words, females are becoming much less fecund as a result of that isolation and that reduced diversity. So this is an example where you have a population that has shown low diversity and a whole lot of signs of inbreeding depression, but effectively is doing fine. There are thousands of them on these islands, so wide volume of genetics doesn't tell us anything. So that's an example where genetics is debatable about in terms of its importance. So the point of the whole series of things I've just talked about is to try and understand how we can actually use that information in wild populations and where we're doing management options in terms of understanding low effective population size, high inbreeding predictions, and then ultimately prediction of extinction. So it provides us with an environment such that we can manage reserves with, and where we've managed all the other problems that go along with population decline. I'm talking about introduced predators, disease, etc., and it allows us to understand that if we supplement them through reintroduction, and we can get a bit of an idea of the metrics in terms of how many individuals we want to be reintroducing to the population, will be sufficient that we can actually maintain, at least maintain, maybe even increase genetic diversity. We just published a paper on Warnings where we've actually shown the genetic benefits of increasing the diversity in translocated populations by selectively trapping different animals over others. So we can use it in even more sophisticated ways to increase diversity, not only just to maintain diversity. Okay, and I'll let you go to that one's there. What I thought I'd do is just give you a couple of examples where we've used genetics and how it's been used for population management. And the first one's quokkas. I think everyone knows that quokkas only exist on Rottnest Island. Does according to the press. Anyways, we're interested in looking at quokka populations to try and understand which ones have diversity, how many populations are there, are we dealing with one standard population that go from Rottnest to wherever they go to, etc. So we are going to ask three questions. I asked a question on, oh, I've got my source of where I stole that thing from. It doesn't say Rottnest Island Authority, by the way. We're interested in looking at population structure. Is there one population or are there multiple populations? We're interested in identifying if there is structure, which populations of those are really important and which ones have shown signs of loss of diversity. And the other one is a little bit of a look at their population trajectory. Which populations are going extinct, which populations are seen to be travelling okay, etc., etc. So these are really basic questions that the recovery team were really interested in knowing about because there's a few things going on with quokkas. And I'll give you one example. They're found on the top of Bluff Knoll, on the top of Fitzgerald. Anyone knows? Does everyone know that area? We have a species that's endangered, living in an area that's endemic. They're endemic to the Stirling Ranges in terms of that area down there. But there are two species of plants in there that are also endangered, and quokkas are eating the plants out. So the directive has been to remove the quokkas from the Stirling Ranges and put them somewhere else. Where do you move them to? Are they the same as other populations? Are they showing more diversity? Are they inbred already, etc.? There's a whole lot of questions that go around removing an animal potentially from that place and putting it somewhere else. Okay, enough talk. So here's the distribution of the quokkas. They don't only occur on Rottnest Island, they occur all the way around from north of Perth. There are historical records, but essentially at the moment they're from the Escarp all the way around through to Albany, Two People's Bay, and there's an island called Bald Island, which is off the coast. And it's very sort of similar to Rottnest, not as big, but it has a very large standing population of quokkas on it. So these are sort of historical are the pink, and the current ones are the blue, or current 1980, some years ago. They're even worse. There's been a new population found down south of Bunbury, so it's living on the Swan Coastal Plain. It's the only remaining population of quokkas on the Swan Coastal Plain. And we're really interested in knowing, are they from this sort of, which population do they belong to, where do they come from? Because there's about 6 animals living in that swamp, and it's on private land, so it's quite threatening. So we had a lot of sampling sites. We collected quokkas from the entire distribution, and we used microsatellite markers, which is the one I've been talking about, to try and look at and determine whether we're talking about a distribution that consists of a single large population of quokkas, or in fact, is it a series of quite discrete, genetically unique populations that occur around the distribution? And if so, what are they? We don't know. And it's not possible to actually look at a quokka and go, yep, you belong over there and you belong over there. Morphologically, they're very homogeneous. So what we use these things called structure plots. I'm just going to spend a little bit of time on these because these are really cool, because we're going to use them later today to look at some questions. And what we can do is we can take those microsatellite markers and we can genotype an individual. And that individual will give us a very unique signature of what you look like. So in a forensic setting, I have a very unique genetic profile that will be used and can be used to identify whether I've contributed DNA to a particular crime scene, etc., etc. And because they're so unique, it's very difficult to sit there and say it could have come from someone else, because they're unlikely to produce the same genetic profile. Now, if I was to take, if I was to take a population of, I'm from Danish origin, and if I was to take a sample of people from Denmark and genetically profile those people, they'll all be quite unique, but overall they will share a lot more than if I was to take the same population sample of, say, individuals from sub-Saharan Africa, for instance. Because clearly they were mixing a very long time ago. Their shared common ancestor was many tens of thousands of years ago. So genetically they've been on these different trajectories, and they show genetic signatures that make them either Danish or sub-Saharan African, or one of the better examples. Make sense, Trevor? If I was to take populations of quokkas, and they're all genetically unique, then they would all be, in this particular case, for instance, they might all be green. In other words, it doesn't matter where you sample a quokka from, it will always appear as a sort of a green quokka. Its genotype is essentially a green one, or a Danish one, if you want to use the equivalent. When we looked at these quokkas, what we found are these, and these are 2 examples where we've used the 2 islands and then their closest mainland sites and compared them to them. So when we looked at individuals from Rottnest Island, we can see, and each individual is a bar. So if you can, it's not a very good example because it's line, but you can see this bar here. Flick forward because I think I'm talking about. Each individual is a line down on that graph, and basically you are essentially looking at how much genetic composition is in that individual related to the site it was trapped in. If I go back to here, if I was to look at, say, this individual here, which has a little tiny bit of yellow, but mainly green. Does that make sense? If you can imagine there was a bar there. Then that individual has essentially got a signature of a quokka that's come from Rottnest Island. And it's got a little bit of a signature of an individual that might have come from the mainland site, but it's not, it's just a variation. Am I making sense? Sort of, right? It's going to get a little bit, yeah, okay. So when we looked at the genetic signatures of all this is what we found. We found that Rottnest Island, quite unique and genetically very different than everything else. There is absolutely no sign of any Rottnest Island in any of the other populations we sampled. So Kesness Swamp, which is the mainland site opposite Rottnest, they're all yellow essentially. Then you've got Rottnest Island. Bald Island is this big red blob, so they are genetically really distinguishable as Bald Island rock quokkas. And then you've got Two People's Bay, which is the mainland site opposite. So what we use something, it's a Bayesian modelling. So if anyone knows about any mathematical stuff, Bayesian modelling is becoming really, really important mathematically. And we use this clustering methods to try and understand how genetically unique populations are. And if we look at these, you can see that this is the break point for these 2 different populations. There's one that's predominantly green, and there's this one that's predominantly red, but there is indications that there are some individuals from that population in this population based on the colour. Okay. And so that's what I was talking about with these structure plots. So, when we looked at these things, this is what we're able to show. Individuals where the parents are mixing between populations is referred to as admixture. And if we had a population where we had an individual in there where one parent came from, say, Kesner Swamp, and the other individual came from Rottnest Island, and we caught that individual on Rottnest Island, so in other words, it would be embedded in here, what would that bar plot look like? Would it be entirely green? No. Would it be entirely yellow? Would it be half and half? One parent comes from Rottnest, the other one comes from Kesner. So in other words, we can pick up the signatures where your parents have actually come from one population and another parent's come from another one. And we can go even deeper than that. In other words, we can actually put together a history of where individuals are coming from based on their genetic profile that occurs. In other words, if we were to take this individual here that was captured in Kesner Swamp on the mainland, and we pick this signature up, we can assume that one parent in Kesner Swamp somewhere has been taken from Rottnest Island and put onto the mainland. And that's exactly what we actually found, although we're not allowed to report that, apparently. So don't write that down. Whereas Wildlife Care has taken an individual and taken it upon themselves to release an animal back into the wild because they think it's the best thing to happen to the individual, when in fact they're mixing genetically different populations together. So an admixed individual will give you this sort of pattern here, where you've got a signature of one individual from one population and one from another. The alternate one is where you've got completely illegally moved individuals. So you imagine taking an individual from Two People's Bay and you genetically profile Kesner Swamp up in the top here around Armidale, and you find that most of those individuals are all Kesner Swamp animals, that there's an individual that's been captured there that's got a completely blue signature. And the assumption, or the only assumption that makes any sense, is in fact there's an individual that's been moved from one population and transferred into another population over there. So this structure analysis is actually a really, really nice and quite sophisticated way of looking at how individuals are moving, how they're genetically unique, how they're maintaining populations, how populations have been mixed or not, and stuff like this. And we've used this in a number of ways, and I'll show you one way we've really used it, which is really nice.
Would that count as migration?
No, that's an artificial movement. So unless that animal's moved naturally between the population, that would be a migration event. It's in fact referred to as an introduction. So translocation is moving individuals from one population to where they used to be, and its introduction is a movement of individuals into a new population where it's never been.
So, like genetically, would it?
Oh, yeah. I mean, effectively what you've done in that situation there is increase the genetic diversity of that population by introducing completely new genetic material into it. And to be quite honest, there may be really good situations where you might say that's actually what we want to achieve. In terms of management, we want to maintain the biggest diversity. Our best option is to actually mix these two populations together. Quite controversial, but I think I'm a big mixer. I think it's a great idea. Anyway. So, what sort of scale are we talking about for quokkas? This is the structure plot that came out of the quokka analysis. And basically, the analysis showed us that Rottnest Island is very unique. So it's got this very brown signature in this particular graph. These are the northern Jarrah forest populations. So they're each quite unique populations. So the northern Jarrah forest are a series of quite discrete, genetically unique populations of quokkas. The southern forest sites and Two People's Bay and Waichinnikup, which is a nature reserve, even though they look different, are actually genetically the same population. So you've got basically populations from Perth down through to Collie that are genetically quite unique. I think I'll let you go. Got a picture, that'll do. So you've got these quite genetically unique populations in the northern Jarrah forest, and then you have this really large single genetic population that goes all the way from Collie all the way around through to Two People's Bay. And then Bald Island's a very unique population off the end there. The reality is that's what that max looks like. These are completely different disjunct populations. So what were once continuous populations are in fact highly fragmented, small, remnant, and in my opinion, these populations are on the road to extinction. They'll be gone in the next couple of years unless we do something really active to manage them. This is a very, This is a very, very busy thing, and I just don't worry too much about the noise. What I want to do is just to point a few things out. I have, here we've got the number of samples we use, so we've got quite large samples we've used in the data set. And here we've got a whole series of different genetic measures between those populations. And I guess the point of what I wanted to show you was, let's just pick one of them, say expected heterozygosity. And this is what the expected heterozygosity is, the number of heterozygotes you would expect in a population of that size. So it's just a measure of genetic diversity amongst a whole range of other ones. But this goes from Rottnest Island, Northern Jarrah Sites, Southern Forest, down through to Bald Island. And we can see there's 40% genetic diversity on Rottnest, so, 40% is not too bad. It's all relative though, because what we need to do is start looking at some of the big sites to compare it to. And the biggest site of crockers that occurs in the southwest is the one that occurs in the Southern Forest. And in fact, it's this one here. So the Southern Forest is 83% genetic diversity. Rottnest Island has about half that. So that's relatively speaking giving you a bit of an idea. And in fact, all the Northern Jarrah sites have relatively large amounts of diversity, but nothing near the 80% down in the Southern Forest. And these populations have similar things here. And then right, get down to Bald Island, has about 25% genetic variation. It's about 500 crockers on Bald Island, but it has a very low amount of diversity in it. So we've got a series of disjunct populations and some very large populations that retain quite large amounts of genetic diversity. They're really important, those Southern Forest sites. And the third thing we looked at was population trajectory. Is it increasing, decreasing, etc., etc.? And this is where I'm going to talk about this genetic bottlenecking again. And these are these sites again. So you can see Rottnest Island, all the Northern Jarrah Forest sites, and these are the Southern Forest sites, the ones down in the southwest, the really secure ones. And when we did genetic bottleneck analysis on these things, we found that all of these populations are showing signatures of genetic bottlenecks. They've all been through recent declines, whereas all the southern sites show no sign of population genetic bottlenecking. So the populations are really stable, maintaining diversity, etc. When we looked at the shape of those in terms of whether there are rare alleles in the populations or not, we see that there is, when you have most populations in the Southern Forest have a very healthy, rare number of alleles, and lots of the Northern Jarrah sites show signs where they've completely lost these really rare alleles and they're flattened out into just really wholesale common alleles. So they're showing very good signatures of genetic bottlenecks. And then lastly, I'll talk about effective population size. And if we look at the effective population size in these populations, Rottnest Island has a very large effective population size, even though it has only half the diversity of some of these other sites. The Southern Forest site has about 65 effective population size, so it's close, but it's still quite reduced. But all these Northern Jarrah sites are in the order of 1 or 2 individuals in those populations, effective individuals in the population, even though there's sort of half a dozen in there. So genetically, these are really an interesting individuals and interesting populations to look at. It's really been very helpful in terms of trying to manage these things. We've been able to identify a lot of these things. There's some suggestion that Rottnest Island has some subspecies occurring on it. So there's a subspecies around the settlement that are different than ones down on the dry side of the island. So we had a quick look at whether there's any genetic structure within Rottnest Island and found there are actually none. So in fact, if we look at Rottnest Island just on its own, if there was genetic structuring, we see them in different colours within the island, but we don't. So Rottnest Island shows absolutely no sign of genetic structuring. So if we look at a PCA, principal components plot, looking at differences, we find that the closest mainland sites way over here, Rottnest Island, are very unique. They're quite genetically unique. They're not anything like mainland sites, but when we blow this up, and there was structuring, this is looking also at geographic distance, we would expect those clusters to come out together, so all the crosses together, all the squares together, etc. When in fact you can see it's just a big homogeneous mess. So crockers are moving all over the island, and they're quite capable of moving very large distances from the islands as well. Okay, so that's just basically a key finding of that. I don't think I'll go into that, but there are lots of different sites. Some of them are quite diverse, some of them are not so diverse, etc., etc. And lastly, I just want to finish off by spinning it on its head a little bit and talking about feral pigs. And feral pigs are at the moment off the playlist, but they are going to come back on. They were introduced by European settlement, so they've been here for about 200 years or so. They became sort of semi-urban. People would have them in backyards, they'd escape, etc. or whatever. There's now something between 5 and 20 million feral pigs in Australia. Most of them are up in northern Australia. We have a large population in the southwest down here, and we looked at the genetics of feral pigs. They are what they refer to as a triple threat species. So they're an agricultural pest, so they'll eat land, they'll eat livestock, they'll push down infrastructure, they'll take out water sources, they dig up areas. I mean, I don't know, has everyone seen feral pig damage? Looks like someone's gone through with a tractor. They're just absolutely extraordinary. And of course, if you think about it, feral pigs live in that riparian zone. That zone that sits on the edges of river systems and things like that, and they dig all this crap up. And then of course it starts raining, flooding and whatever. What do you think happens to all that stuff? Off to Mecca. It's just gone. So the damage they do is unreal. And the other one is the diseases they contain, or they can contain, sort of blue tongue, foot and mouth, sorrow, just everything you can imagine. Pigs know the whole lot. So they're one of the few feral pests that have an effect on each one of those things. Things like feral cats are of course predators, but they don't trample the ground such that it changes and things like that. Feral pigs do. So we thought, why don't we use genetics as a de facto tool to try and understand what feral pigs are doing in much the same way as what we were doing with quokkas, to try and understand how big are these populations, where they move, how being affected population sizes, etc., etc. And the way we used this in the most creative way was we identified that there were 7 feral pig populations in southwest of Western Australia. There's one up in Naroma, up in near Northampton, and then there's a series of populations that essentially equate to the land water system, so the drainage system. So when you have a drainage system going into a dam system, that's where a pig will set itself up, which makes a lot of sense, and they tend not to move between the hills to another one. And the way we were interested in using it is to do things like, and we'll do this today in our lab, where we'll look at the population structure using principal component type plot. And we've identified that there are different populations. So you can see here's a green population, red population, etc., etc. The only standout is this individual here. So this individual here contains the signature of that population over there. In other words, the only explanation for that, given everything else, is that that individual is in that population because it's been moved from that population there. Feral pigs, imagine going into a population and sitting there going, I think that pig moved from there. There's just no way you'd be able to determine that. There's no way you could do that. Yep. So what we did was we identified these genetic populations, and then we looked at using this population assignment, this graph, this bar graph stuff. Is that right? Right, stuff that. See this graph? All populations on the bottom here, and the genetic uniqueness of it on the x-axis, on the y-axis here. Each population has, this population was taken from Denbarker, and 100% of its genetics is from Denbarker. Right, so they're Denbarker pigs, and they share no genetics with these populations here. Sorry about that. So if we look at that, all the individuals in Denbarker, where they all are. Then Perth Hills, what we identified was that there are these 3 individuals here that were genetically unique to Northampton, Port in Perth. So they've been illegally moved by recreational hunters because the population of pigs has declined. Pig hunters go in there, capture a pig, dump them off into the forest, and the next time they go out there, there's pigs enough for them to go and shoot them over there. And I'll leave it at that. Thanks, folks.